Hello everyone, welcome to the third in our SCI webinar series. I'm Nessa Carson and this series is produced by the SCI Fine Chemicals Group and the Young Chemist panel. So a little bit more on those two panels. We exist to facilitate links between organic chemists both in industry and in academia. And it's kind of just a forum for discussion and sharing of information about pure and applied synthetic chemistry. This mostly is in the form of running conferences and other events. So I'll be chairing the webinar today, and alongside me are Fred Hancock from the FCG and Ollie Ring from the YCP, who are going to be moderating the questions later. So about questions, then please ask a question at any point during the talk. You can do this by typing your question into the chat box. You'll probably see that everybody has been muted other than the speaker at the moment. And we'll then put as many questions as we can to the speaker at the end of the presentation. Well, without further ado, then I'll introduce our great speaker for today, David Lathbury. So Dave was the Assistant Director of Process Development at SB. And then he moved on to become Head of Process Chemistry at AZ in Lathbury, before becoming VP of Chemical Development at AMRI from 2010 to 2014. Since then, he's been a process chemistry consultant, and today he's going to talk about early CMC studies in drug development at the medicinal chemistry and chemical development interface. Over to you, Dave.
Thanks, everyone. Very kind invitation to give this talk. As you'll see, a chunk of it actually has been published 2 or 3 years ago, maybe a little bit more, as a chapter on chemical development that Dave Ennis, who replaced me at AZ, and I wrote, and is in all good bookshops. I think Dave, I was talking to Dave the other day, and he said there will be talks about updating this. So some of the stuff that I'm going to put in towards the end might also find its way into the talk. So one of probably the only advantage of getting older is that you can get a perspective of how things have changed, and also sometimes how things repeat themselves and how they stay the same. So what I'm going to talk about today is to a large degree focused on smaller companies, companies that don't have any chemical development or very little chemical development expertise in their companies. And the interface, therefore, if I went back to my first early career, then these departments were separate, finite, definite departments with organizational structure, and that interface to some degree felt a little bit like a wall, and often once or twice a year a bunch of chemicals were lobbed over the wall and we basically just took care of stuff, and then Medchem went back to make the next lots of chemicals to chuck over the wall. Now that's changed significantly. A lot of these companies are virtual, they don't have any chemistry in them at all, and that then puts a bit of a hard wall between the sponsoring company and the actual chemistry that's being carried out. So no one in the company that I'm dealing with potentially has any hands-on experience with that chemistry. So in many cases, that interface now is not between my client and me, it's between my client CRO and me, which makes that even a little bit more distant. And you'll see these studies in many cases are virtual in terms of their thinking, punctuated by a few experiments, but a lot of those can, if they're carried out early enough, and this is the key, is doing things early, don't wait before they become an issue, can certainly mitigate, in some cases completely remove issues that are going to crop up later on that are going to cost several months, if not a year, and potentially cost you an extra several $100,000. So what are the differences, chemistry and chem dev? And I think these have always been there to a degree. There's obviously the scale. I think the scale at which Medchem operates, certainly for analogues, is significantly reduced. Showing my age, but when I first was in Medchem, or doing my case award at Glaxo back in a few years ago, that you couldn't get into the screens until you've made around 300 to 500 mg of a compound. This will get you halfway to candidate selection, maybe more on this with certain potent compounds. So scale is important. That's the big difference. Reaction times, therefore, things take longer, quite a bit longer. Process safety it's still there in medchem, but the consequence of getting it wrong, given the smaller scale, is less of an issue. And that also comes to compound toxicity and things like that. The two biggest ones, I think, are very different. Analytical, there's a huge amount of analytical in chemical development. I think it often surprises medicinal chemists exactly how much. And things that are actually gradually, certainly at the analog stage, are almost drying up and dying out in discovery. And it's no one's fault. It's just a consequence of lower and lower scale and technology, workup and isolation. But in fact, workup and isolation actually occupies probably around 70% of chemical development's time. So making the bonds is often, not always, but often the easy bit. Getting the material out pure enough in a high enough yield at the right, say, crystalline form is actually where most of the work and most of the time is spent. So the project came in, lobbed over the wall, you caught it, and at which point you'd be just looking at that molecule. And the two things that you'd be looking at from a phys props point of view, stability and solubility, things like melting point. Was this thing compatible with the sort of mode of administration? So, you know, if it's 0.2 mg a mil and people want an IV drug, then there's going to be some issues. If the solubility is low, and I'll have quite a bit more to say about solubility, or it's got a very low melting point, obviously these things are just not compatible with modern day formulation, and we're going to have to change things. And if we can figure out those changes ahead of time, then they don't necessarily have to be on the critical path. And if you take nothing else away from this afternoon, I hope I'll convince you to some degree, is that a bit more thought earlier on and just asking a few more questions can really save yourself a whole load of trouble. So speed and cost of API production, I'll say quite a bit about that. That's important, I guess, for several things. When I first started in big pharma, it depends on the complexity of the compound, but for complex compounds, it can have a big impact on the net present value of the project. Is it financially viable? Are you going to make a profit? Nowadays, for small companies, it's a lot of these deadlines are driven to funding rounds. So if you can get into first in man, you'll get your next tranche of funding. And you know, this is a big issue. You know, it's potentially existential. But as I'll say and probably keep repeating, and I'm not decrying the importance of this, far from it, but actually nature doesn't care. So, you know, when do we think about these issues? Now, lots of companies have different models, and I think we do think about these things a bit earlier, but I think sometimes we don't think about these things early enough. When things go wrong in a drug project in the early development, certainly in the CMC point of view, if you look back, there are almost certainly clues earlier on that would have actually helped you solve those problems, and we just didn't take any advantage of those. So cost of goods, I'll say a little bit about cost of goods. Although it kills a small but not an insignificant amount of compounds in the longer term, it's hard to be definitive. We don't know the dose, we haven't got a manufacturing commercial process, and we don't know the volume of the production. And volume, in terms of using commercial to drive down costs of intermediates, is purely dependent on how much drug per year you're going to make. If you're making a compound that's going to be a ton or less than a ton a year, it's going to be very hard to get purchasing to drive that cost down. Having said that, I've only seen 2 drug candidates in my entire career that have been killed at birth before they got into development. And we knew quite a lot about that area, both beta-lactam antibiotics. And I think I could quite confidently say at that time that something at 34 steps, 0.1% overall yield with a target price of $2,000 a kilo wasn't actually going to make it. So I didn't feel so guilty about killing those. So if you're sat in your project team meeting and clinical are giving you their life story and you're a process chemist, all you want to ask is 2 questions: how much and when? How much API do you need and when do you need it? Again, it depends, doesn't it? It depends on what you're trying to achieve. You could be with a project where you're actually wanting a very quick exposure into man to get some PK information. And then you're potentially going to come back and follow up with a better compound, or you've got some real issues. For example, I can remember a project where we had about 3 or 4 prodrugs. And so the initial bit was to get 4 compounds into man and just see which was the better prodrug. You could be a company that's taking a longer term view. So maybe, heaven forbid, doing CNS. Not many companies do that now, but you may have quite a late stage proof of concept. So you're going to be making a fair amount of drug. And so you're taking a long term view. How fast are you trying to move? As I said before, have you got a really important funding event that you need to hit? What level of contingency do you feel happy carrying in the system? You've got to carry some. When you look at how estimates of drug substance are made, it's quite frightening. Then some of these have virtually no contingency in whatsoever. Now, contingency is an important thing because you've got to make sure if you're a project manager, you carry one load. What typically happens in the past is that, say, formulation want 2 kilos, they'll ask for about 2.5 or maybe even a bit more. And so we're given the right, you need about 2.5 kilos. So we'll say, well, we need a bit of a contingency on that. So we'll make another 5. So we've got 3 kilos we're making when we only need 2. Now, that's not necessarily a problem. In some compounds, cheap start materials, easy process, that's not a big deal. You might just as well make as much as you can, as soon as you can. But in certain compounds, it can be really important as to whether you need to go back and repeat something or we can actually carry on. And so the project manager needs to, A, have a contingency, but understand that it is one and know exactly where every last gram is going. So depending on the individual projects, it can range from 800 grams or so if you're doing a single dose in man to over 10 kilos if you have a compound that is not particularly, is quite well tolerated and you have to dose up to 1,000 mg per kg in dogs. Cost of API, order of magnitude, probably about $300 or $400 to $1 million, okay, for a multi-kilo delivery. But there are huge variations, and I think you know, on average, it's probably, I think when we looked at this across all of our drugs that we'd looked at, it was around 3 FTEs and $400,000. So it's not an insignificant investment. So how much? Again, really difficult to answer this, but there are probably clues within the project, certainly to give you maybe upper limits. The initial biological evaluation, maybe you've got an oncology compound that's quite toxic, so you know you're not going to go up to these heroic doses. You'll probably go up to something approaching the therapeutic dose. There might be some related compounds. I gave you the example of beta-lactam antibiotics. That was towards the end. One of the last beta-lactam antibiotics to come into SmithKline in development, and we had a huge history to rely on, so we were quite confident of the potential dose. If you haven't, then what we used to use at AZ was some rough template, and this was based on a whole raft of drugs that had gone through development. So we were saying originally for some rat 5-day, 7-day rat tox, some initial solid state work and a few other things and DMPK and stuff, 50 to 100 grams, 150 to 300 for dose range finding and maybe further solid state work, and then somewhere between 2 and 5 kilos for 28-day safety for an oral drug, with dog being the second species. And even at this stage, you can look at your synthesis coming through, even in medchem, fairly early on, and just ask yourself the question, okay, we don't really know anything, but these are not atypical amounts, I would say. And how do you feel with your synthesis? Do you think it would cope with that? Or does this absolutely give you sleepless nights? So it gives you sleepless nights. The earlier you can understand that and the earlier you can do something about it, and you'll sleep better later on. If you just sort of say, well, it's too difficult to think about and just we'll worry about that later on, I'll guarantee you're going to have some problems later in development. So when do we need it? Well, again, how long's a piece of string? You know, how fast do you want to go? The two main things, probably the three main things, is how long the synthesis, lead times of key raw materials. That is a key thing, and that probably impacts projects more than any other single thing that I deal with. How good is the chemistry? And what I mean by that, is it particularly reproducible? What's the sort of cycle time? You get a feel for, could we do that on a, you know, just scale that up, or is it very tricky? On average, so we're just playing around with numbers and trying to work out, you know, lengths of time and when is a good, you know, when do you think you'd be in the clinic? You've got to plan on between 2 and 4 weeks per step. I would normally start off initially at 4 and see what that timeline looks like. It's funny, this sort of, as a rule of thumb, when you go back and look at historic data, most projects land in that it's one of these universal constants. We're not really quite sure it's true, but it seems to be quite routine. And something I'm sure all good project managers do, but if you don't do it, you should start, and that's plan front to back and back to front. What I mean by that, so if you've got a first in mandate given to you by your investors, you start working back. Say, well, if we've got to start in the clinic at that stage, safety needs to be completed by then, which means the tox batch needs to be made by X. Okay, so you get your theoretical plan as to how you might get to that first in mandate, and then sort of reality kicks in. And you say, standing from where we are, how long do we think it'll, starting from now, how long would it take us to go forward? And I'll guarantee that the two plans don't marry up. And it's that realization of, so how can we get them to actually all, changing things around and maybe starting a bit earlier, cutting some things back. So you finally get a plan that actually matches reality. If you think it's going to be a hard effort to make your initial kilos, and you'll know that, from your experiences in medchem, even from your contractors, that if you quiz them the right way, you'll find out. You've got to think very hard about booking tox slots before you have a very good idea of the API. There can be some pretty large penalty fees now with tox companies if you miss your slot, and especially if you get to the stage where they've already ordered the animals. And again, I get, as I said before, that some of these funding deadlines are really true. They're existential, but as Richard Feynman famously said, nature won't be fooled. It doesn't matter how good the guess, how eminent the guess are. If the guess doesn't match experimental, then the guess is wrong, and you will be found out. So things that impact speed. I could have put a huge number of slides together on this. There's almost, how many project managers in the audience actually figured out that C19 was going to hit back in November? I suspect not many. So the main take-home message from this slide is at least 1 to probably 3 things are going to hit your project. So I'm a real fan of Margaret Heffernan, and she has this good thing about efficiency versus effectiveness. And if you've got no white space in your project plan at all and no contingency, I'll guarantee, absolutely guarantee, put my mortgage on the fact that you will be late. Something will happen. And depending on how hardwired, if you've booked slots and things like that, you could end up losing several $100,000, and then you've got to go back and try and recapture your next slot in tox, which is sometimes booked up quite extensively. It has been recently. So you can easily blow a year worth of development time if you're not careful. What's the main thing in terms of drug substance? And this comes back to a question I was once asked when I was at AstraZeneca. Someone said to me when we were doing capacity management, how many drug candidates can your group take? And of course, it's a stupid question, as were probably most of the questions I was asked by management at AstraZeneca. It's not number of compounds, it's number of chemical stages. That's our unit of capacity. So if it's a 5-step synthesis, you can do X. If it's a 25-step synthesis, you can do 5 divided up by X. You're not going to be able to do that many compounds if they're 25 steps each. And that's because the same in terms of some degree with medchem. Chemistry is still largely an experimental science. We're getting better every year incrementally at developing our theoretical basis, but we're not that good at predicting. And when you come to things like workup and things like how is something going to purify, and is that impurity going to be taken down by the required 0.2%, I will absolutely guarantee you can't predict that. You have to do that experiment. And so, the step count is a very good measure early on if you've got nothing else in terms of how big a job have you got. So this is an old slide that I used actually back when I was in AstraZeneca to try and educate senior management in terms of the consequence of step count. So you've got Brilinta, which was invented in Loughborough, 24 steps in total. I think there's 15, 16 linear steps when it first came in, longest linear sequence. And we've got compound A, which I think was a CCR3 antagonist, which was 6 steps long. Okay, 2 compounds, obviously one looks a bit more complicated than the other, but you know, in terms of the main differences were the actual number of chemical stages. So what does that mean, the consequences? And I'll put all these rather than build the slide. So delivery. So compound A, we managed to, I think it took us about 7 months to make compound A in total, from doing the lab work and then going through the plant. We delivered that pretty much at candidate nomination. Brilinta, we took around 6 months more to actually deliver just under 1 kilo. That was a huge team effort. And the why it was only 0.85 is that luckily for Brilinta, the second top species was marmoset. I often reflect that if it actually been dog and we'd have had to make something like, I don't know, 5 to 10 kilos of Brilinta, it would have taken us so long that, given that Exanta was still on the market, was coming onto the market, I wouldn't mind betting that Brilinta would have been canned or sold off for portfolio reasons. So in terms of how do you get to the final route of manufacture, what we would call in the old days synthesis freeze, the final route for compound A was actually all in place by the time we'd made 28-day safety material. It was a very easy compound to make, and we pretty much decided that's the best route of synthesis. For Brilinta, that final route selection, and that was the high-level final route, there was still more details to thresh out on some of the intermediates, that took almost 2 years to get to. So the other interesting thing is also the amount of resource, and this is because of the project management system and the resource counter. You could easily get hold of these figures quite quickly. We needed 2 chemistry FTEs effectively to get to route selection for compound A. We needed 24 FTEs to get to compound selection for Brilinta. So I'm not saying, and this, I think the audience misunderstood this when I was actually saying this. I wasn't saying we want simple compounds, far from it. You have to absolutely select the best compound that you have. But step count and API amounts can have a big impact on speed. So if you're going to pick something complicated, because the biology says it has to be that compound, you obviously have to know that there's consequences in terms of when you can hit your targets. However, unlike some other things, you know, route development can often alter that delivery situation pretty quickly. Let's do a little bit of chemistry, and if we've got time, I've got a few more slides like this towards the end. So this is what we call inefficient chemistry. This actually was done by medicinal chemistry. It was done in Lund, and it was done by medicinal chemistry because at that point in time, I think we had Crestor, Iressa, Thiazan, and something else all in my group. So everyone was pretty much working on phase 3 NDA candidates coming through to filing. So we literally didn't have a single chemist to pick any of this up. So you'll see the chemistry looks relatively straightforward, except when you get to the end, you've got an awful chiral chromatography. And from a medchem discovery point of view, this makes total sense. There is absolutely no point in spending time trying to work out different asymmetric syntheses for things that are not going to last more than a couple of days once they go into screens. So this was fine. But obviously now it's not fine, because now we're looking into making sort of 10, 50, 100, maybe 200 or 300 grams. And in terms of the costs needed to do this, this just wasn't going to hack it at this point in time. So we had to have a change. And so we got together with the guys in Lund, probably a couple of hours, and we very quickly came up with the synthesis you see here. So obviously the way to think about this is to break this sulfonyl bond here, so sulfonyl chloride and the piperidine. So then we're into the Reberg synthesis, which we knew how to do. This being now a much more lipophilic and a much better separation, we could separate that on easily on chiral chromatography. 45% is, the 50% is maximum theoretical yield, obviously. We do the oxidation to the sulfonyl chloride with chlorine, and then we bring in this second half, this piperidine, and then couple up. So you'll look at that, and that's a 27% overall yield. So that's gone from 5% down up to 27%. So this actually illustrates a point, and it's something I've used in many cases to try and explain why changing the route isn't necessarily going to add to a delay. So if you've got a 7-step medchem route, and the figures on the right in the table, 18 weeks, 6 weeks, these were, one is the actual are the models that we got from. We had a throughput model that was actually pretty accurate when we were looking at various routes. You could actually predict, if you knew roughly or estimated what a maximum volume would be and a cycle time, you get quite accurate figures of predicting how long things would take in a kilo lab. But even if you take the medchem route and we say, right, we haven't got time to figure out something better, you're still going to have a load of work to do just to optimize that, to process safety, evaluate that, to develop the analytical methods. You know, there isn't effort free. You can't just walk in with your sequence and write, bingo, you've got compound. So that means you can spend a fair amount of effort on the new route down here in terms of coming up with something better. But as long as when you do this, it actually shrinks this bit. So obviously we take 4 steps, almost chop the synthesis in half. So it's going to be nowhere near as much development work. It's going to be nowhere near as much production work. So the way to think about it is that, yes, it is an additional activity, but it shrinks the related follow-on activities. And therefore, theoretically, it is quite feasible. In fact, in most cases, almost certain that if you come up with better chemistry, you know, you're going to significantly alter and improve both your time and cost. So the outcome. So this is where I think, and again, a big take-home message. Initial lab work to demonstrate that chemistry took less than 2 weeks. And in my experience, this is normally the case. If you put a bit of a concerted effort in brain power and just a bit of focused lab work, things break quite quickly. You get improvements pretty quick. It's very rare that you're bashing your head against the wall for, you know, 6 months with nothing. And the consequences of route improvement are really stark. Discovery then developed 40 grams for initial tox, and they actually delivered almost 300 grams in 4 weeks compared with about 18 weeks that was predicted. The last thing is quite surprising. I think initially it surprised me, but actually when I think about it now, it doesn't. So the reason I use this slide and still use this slide is that the original route was sent out externally initially and costed. And when the cost came back, I think it was Astute that costed it. Lars Erik Arvidsson, who was the head of chemistry at Lund, almost fell off his chair and said, like, we can't possibly afford that. When we got the improved sequence, we sent that back to Astute, who costed it again. And actually, for the first GMP manufacture, we would have saved over $700,000. And that's a staggering amount of money. It's not atypical. It's probably on the high side, but not necessarily. And I would say that most of the time, when I've been caught out with stuff, it's not because we've not been brave and we've got caught out trying to change the chemistry. It's normally when we've not been brave enough. So physical form. So that was my message for the drug substance. Be brave, be early, use your CROs. You know, a lot of them are creative. You know, if they're doing chromatographies, try and quiz them a bit more. How bad are those chromatographies? Are they even remotely possible on scale? And the moment you get to the stage, you think, actually, we can't scale this, I would really invest a little bit, even if it's a week or two from one of your CROs, to try and see what else they could come up with. Most of the ones that I work with can do that. Even in the medchem ones, they can do that. And there's nearly always some really good frustrated process chemists in that group that can actually do an excellent job. So take advantage. And if you do it early, you've given them maximum time to be successful. This form, this is another thing that surprises me, that quite a few programs, and I don't know why, often put this off in terms of understanding this right towards the end of the program. And although to do this properly, and you'll see the amounts I've got on a slide in a minute, you do need a reasonable amount of material. You can do stuff on the fly and get you some good heads up information without using that much. So there are 2 key questions. Can formulation use it, but can ChemDev make it? You know, ChemDev cannot select a form that a formulation can't use, but formulation can't select a form that ChemDev can't make. It's as simple as that. There's not an either or. It both have to agree with this. Also be aware, we talk about this in terms of clinical formulation. Tox formulations have become more of an issue. What seems to have happened, certainly certain tox houses have actually significantly slimmed down their formulation development capability. So be aware of that. If you've got something that might need a bit of an enabling formulation, and you might think, well, the tox house have got expertise in this, just be a bit careful. We've been bitten a couple of projects where we've had to take this out, give it to a separate group to do a tox formulation before we can send it back and do the tox study. But again, you can check these out way ahead of time. You don't have to wait till the last minute. So the fizz form, well, what would you do? Solubility, stability. Could, just getting some either aqueous solubility. You don't have to necessarily get a full dynamic solubility. You can measure this if you can find an HPLC. Stability, you could stick it in a little vial in the dark or in the light and just leave it a week, get an NMR. Does it decompose? You're looking for gross changes here, and they're not particularly accurate, but they're better than nothing and better than ignorance. So even if you find something, you find, well, it's really soluble. It probably means you're okay. It might be an amorphous form. You've not really checked out whether it is or not, but it may be okay. If it's really insoluble, and the chances are it may well be amorphous, it's not going to get better than that unless you do some enabling work to it. So right there and then, that's something you've got to pay attention. Now, you can decide not to worry about it till a bit later on, but at the very least, you've got to carve out some material to fix that, and you've also got to carve out some time within the development plan to make sure that it gets fixed in time for it to come through to the final API. Early tests, but they don't really take a lot of material now. Technology's moved on. A quick initial solubility, stability screen, and looking at crystallinity, probably less than 50 mgs. Now, I know early on you haven't got many compounds with 50 mgs, and that's a lot of material. But actually, as you move on towards compound selection into later LO, you can get these amounts of material, or if not, you can specifically set out with either internally or with some CROs to get this material. And trust me, it takes a lot of pressure off in terms of the timeline if you can actually identify this as an issue. So, for example, if it's a real brick dust candidate, you can still make them. I'll say a little bit about that in a minute. But you can't. You can make them, but you might need to do something for the safety assessment. So it's not just the dose of your compound. You're going to have to get way more than that concentration exposure in tox species to prove that you've got some sort of tox margin. Sometimes 10 up to 100-fold, maybe even a bit more you need. So, you need to get much more solubility for your tox formulation than you necessarily do for your clinical. So, for example, if you decide you're going to go for an amorphous solid dispersion, you're probably going to need something 50 to 100 grams. And, it could take you several months, even to get into the CRO. They're probably, in many cases, booked up. I think that's a key issue. One of the reasons for thinking ahead is just to be able to secure a resource. If you decide, right, we need to go now, let's go and approach a company, say Onyx, don't be surprised if they can't pick it up for another 3 or 4 months. If you'd known about that 3 months ago, you could have secured it, but you've basically now got a 3-month delay, and there virtually is nothing you can do about it. So a mini salt screen, once you've maybe got a compound you're more interested in, 1 to 5 grams, and if you talk to these companies, they will probably do the 1 gram or a bit less. They'll probably be able to do certain significant work. Although a full salt polymorph screen, which I would only advocate doing on something you believe is a genuine development candidate, that's around 20-30 grams. I think be careful in selecting the CROs is something also that I would see. Again, this is an area where if you've just been in medchem all the time, you may not have got a huge amount of exposure to. But selecting CROs, in my experience, and there may be people in the audience who will vehemently disagree, but that's fine. I would prefer a CRO that's an API CRO with a good solid form, that they understand, how to make it as well as select it, and they can also be very good at developing a good crystallization method. Because until you've got that, even if it's a great formulation, you haven't got a project. So, analytical. This is an area of big difference between medchem and chemical development. And I think this comes as quite a surprise to quite a few people when they first get into this sort of area. To develop a decent HPLC method that's stability indicating, first of all, you have to do a force degradation study and optimization. It could take you a good few weeks, in some cases quite a bit longer. I've got a particular project at the moment where this is actually the issue for the project, trying to develop a decent, reliable stability indicating we've been at this about 3 or 4 months, and it's still not fully cracked. To release a batch of drug substance out of the plant for the clinic takes at least 2 weeks, in some cases 2 to 4, depending on QA sign-off and things like that. But early on, and if you're starting to get batches, small batches of compound that you're doing little bits of tox tests, you can use NMR. It's quite good, especially if it's a relatively simple NMR. If you've got 1 or 2 key peaks that you can look at, you can look at some additional orthogonal methods, TLC, mass spec. You can get these Amicad TLC readers. You can almost get quantifiable TLC. So there are lots of things you can do to at least to get a handle on whether this thing's stable, whether you're making, you're making tests and making recommendations on compounds you really don't have a good idea of purity. At least it will give you an idea of relative purity. Virtually free, and you never know. You might, you do a stability test on something, it doesn't look that stable, but then you find out it's only 90% pure. When you get this thing to close to 98% pure, actually stability is fine. Let's talk about quality. This is something that is, again, something that will be alien to a lot of people in the medicinal chemistry side. One in particular area that it will come as a bit of a shock are potential genotoxic impurities. Don't ask me where this came from. In my view, this is not a patient safety issue. It's a regulatory BS issue, but we have to deal with it. And if you don't take it seriously, you just won't be given, you won't have your INDs or IMPDs approved. So there's lots of reference and guidance. I've given you a couple. There's a book by Andy Teasdale, who's probably one colleague of mine, ex-colleague at AZ. Andy probably knows as much, if not more, than anyone else globally about this issue. He's been working heavily with the regulators to introduce decent guidance. He's got a good book. There's also a paper there, which is a joint paper with Pfizer, I think Pfizer, Glaxo, and Abbott. And there's also M7, which is the FDA guidance on PGIs in about 2013. What would I say about this is don't panic, don't automatically redesign the synthesis to try and solve this issue. In many cases, you can actually use crystallization or lots of other things to actually get rid of these things when you see them. You have to do a theoretical assessment of what reactives could be in your compound. It's hard to do chemistry without using electrophiles. So most of the things that you'll use, or quite a few of the things, including some boronic acids, will flag on QSAR as potential mutagens. And the other thing to remember is if you think if it is a PGI, potential genotoxic impurity, you have to treat it as if it is a GI. So in which case it is guilty until proven innocent. So if you've got a chance and you've got it find it hard to control, then get an Ames test. Because if it's Ames positive, you haven't lost anything, but if it's Ames negative, you can treat it like a normal impurity. But key here, and I think key to a lot of these things, is discuss this with safety assessment. Don't just go off on your own and think you've read the books and you can take care of this, because it is a lot more complicated, especially compounds that flag as ambiguous within Derek and say lead scope. You need a detailed tox assessment to actually sometimes tease these apart and make a decision. Another myth, GMP material made to GMP is only required for clinical studies. It is not, and I repeat, not required for safety or a GLP tox study. It's not required for formulation development. Now you can ask the question as to, and I know at AstraZeneca we did this and still do this quite commonly, you make a single batch to GMP and that batch does both tox and does safety. The reasons for doing that: one, it's just one campaign, it's often cheaper, it's often not much more time, if any time extra. And the other main thing is the fact that it's the same batch. If you've missed something in terms of you haven't picked something up as an impurity, it's automatically qualified because it's the same material that went into both studies. The number of steps that you have to do to GMP, that's a bit subjective. I think typically 2 to 3 seems to be the general thing now. Some companies, for reasons that are beyond me, do 8 or 9. There's no reason for it, especially early on. This is for early phase material. This is not, and I repeat, not for commercial manufacture. You could well get pushed back quite a few stages on the commercial route, and that's the subject of a whole talk in and of itself. So there are various guidelines. You can see them here, the guidelines for the IND and the FDA web pages. There's some good references there. You've got the ICH web pages. I would draw your attention to the one at the end, ICH Q3A C&D, which is around impurity management, solvents and elemental impurities. Be aware though, ICH is around marketed drugs, and they're based on a lifetime exposure of 10 grams a day. So you don't have, if you hit ICH levels, you're absolutely fine. You can often, very often, get significantly higher than ICH levels in early development and still be okay. But as a starting point, I think it's really good. It shows you the sorts of solvents that are recommended for late stage chemistry and the sorts of solvents that are probably not recommended, and certain ones that you wouldn't go anywhere near, certainly anything that's going into the clinic. Medchem, tech transfer, what helps? I've seen all sorts of manner of things, from a few notebook pages to some things that are absolutely excellent. One of the things, especially, are things like samples. Even TLC samples may help develop an analytical method early on when you're going into sort of tox and GMP studies, especially if you're trying to develop a decent method that can make it able you to compare the relative purities of the various batches. So the tox batch, or sorry, the GMP batch has to at least match the impurity profile of the tox batch. In most cases, people like to feel comfortable that it's even purer. So TLC samples can help, but if you've got intermediates, that's even better. There's I'll give you a little tale, a compound at AZ where I think it was about 8 steps to a key intermediate. And when it came into development, all the issues were around this final step. You know, the yields were appalling, but we thought, well, if we can get that cracked, we've actually got the base of a good method. But a week before that, Medchem had something like 30 grams of that final intermediate, and they blew the whole lot. They didn't need to, they just didn't even think. And it meant we had to spend about 6 or 7 weeks slogging through, getting about 50 to 100 grams of this intermediate before we could sort out the final step. So that was 2 months just blown, but you know, just for a minute's thoughtlessness. Obviously references, I mean, including patents, any ideas, observations around solubility of intermediate exotherms. Again, this is very hard, I guess, if that chemistry is being done remote. As things are getting more towards, you know, the late LO, I would encourage you to ask more and more questions and be quite forensic about the chemistry that's coming out of your CROs. The other thing, as I said, mentioned before, sourcing of intermediates. When I was younger, I thought this would all be sorted out. I thought we'd have one database, you put in a structure, and lo and behold, a supplier would pop up, and that's not the case. Trying to trace, I think when I was at AMRI, we spent about 9 months trying to figure out where a source of thiophosgene was actually coming from until we found it. Proper detailed experimental write-up with actual range of yields obtained, not just the best one. And in that write-up should be the experimental workup and isolation, including solvent volumes. You know, if you've measured it out, you can record it contemporaneously as you're doing it. It shouldn't be extra work as long as you're doing it at the right time. And so all these questions, or the answers to all these questions will help us say, well, what is our route that we're going to use to make the first campaign, the first either GLP tox or the GMP campaign. Quick points about outsourcing. Lab and plant capability, so CDA, all these things. So, you know, I've seen sometimes 2 months wasted by secrecy agreements. Lab and plant availability, I've just mentioned that. Don't assume that, you know, there are loads of labs and staff readily available to take your project. Most of these companies, and certainly because the margins in this sort of area are significantly lower, although they've improved a little bit, but significantly lower than they were, say, 20 years ago. Just for interest, when AMRI started, I think about 1990, they were able to charge $300,000 a year in 1990. So in real terms, I'm not quite sure we've even got to that now, 30 years on. So, you know, if you think, even once you start to think about it, start talking to these CROs. They're fine. They know that lots of compounds might not make it. They know things will be delayed. That's the world in which they live. They fully get it. They're not going to be annoyed, but you'll be annoyed if you ring up and they say, basically, that's fine. This is April, but we can't actually start in the plant till October. The other thing is around sourcing raw materials. If you're now going to be late till October, and you've got a 6-month lead time, so for your first intermediate, unless you've got any around, then you basically can't start until that material comes in. But again, they know, all good CROs will know about these. The important thing is, does the capability of the CRO match the scientific challenge posed by the project? Some of that is, some are better at plant work, some are better vessels, some are better analytically than others. But this could be something as simple as, if it's a very long synthesis and the CRO doesn't really have any vessels bigger than about 50 litres to 100, that might not be the fastest way of doing it. And they'll tell you, if they can't do a good, they will tell you up front. But then think about supplier. You've got to get a tech transfer package. The better it is, the quicker they can start. And the more samples you can provide and the analytical data, the faster they can start. You've got site visits, you'll probably have some audits, there'll be various agreements, look up the lawyers, and cost negotiation. You can blow 6 to 9 months doing this stuff at the end. So if that's the case, it's almost criminal now to have this being done right towards the end. You should try and get this done offline where you can. And even if it wastes a little bit of time, if it's not on the critical path, then it's time well spent. So the conclusions. This interface is important. The fact that it physically doesn't really exist anymore in many industries. It's not the department. It's virtual. It's in the air. So it's really for you to engage sort of mentally with this interface. And I think most of the med chemists I've dealt with that have actually got involved in this and come through this have really enjoyed it. And it is, and I've learned a huge amount from this. But if you can get these questions, just posing it, knowing that you've got a problem is much better than thinking you haven't. The other thing is to think about ChemDev has got to be a phase ahead of the rest. So why is that? Well, that's just obvious. Before you can start any formulation development work, it means you need API, which means that process needs to have been kicked off several months before, which means the route needs to have been placed, but it's such that we know what starting materials to actually order. So if you don't think about these things and you've got a long lead time for starting material, you've blown your timeline. The other thing I forgot to mention, actually, talking about this, we did a bit of a modeling I did with a finance guy in AZ, and we worked out that if you lose up to around 6 months to a year of development time, you potentially can knock off up to 30% of the net present value of the project, and that will definitely impact the amount of money that you're given for your project if you're a small company. When I've done enough due diligences, and really the only thing that they really wanted to know, they didn't care about the chemistry, they just figured out that we'd fix it, and not unfortunately 99 out of 100 we did. But what they were really interested in, when we're going to file, when's the file date, and when can be the launch date, because that then governs how much money that's worth. So things like certain PRD skills. So I think, if you've got a decent analyst near your group in MedChem and you've got an HPLC, start using HPLC, especially for the later compounds. You get a lot more information. Communication is key, especially now we've got a lot more CROs. It's a lot more virtual. I'll say a little bit more about law here. It's just like folklore. Appreciation of PGIs, if you're getting involved with tox batches and clinical, is a must. And if in doubt around whether you should do something a little bit speculative, apply the Grabowski axiom. Ed Grabowski was a good friend of mine. He was a former director, vice president at Merck Process. And Ed's axiom is the following. If you're a chemist and you've got a good idea, but it's a bit zany, just try it. If it doesn't work, just keep quiet. If it does work, you're a hero. So what I'll do is I'll whip through a couple of more chemistry slides, and then we'll just end. So I thought I'd just give you a few more examples. I gave you one about improving chemistry. And just to show that this isn't a myth. This is actually, this happens, and it can have huge impacts. So this compound here, this tetralone amino acid, we needed about 2 kilos to make some various derivatives. And when you look at the medchem route, you can see that actually, although again not particularly steppy, but the yields were just horrendous. The workup, I think it was about 2% overall yield from a tetralone. So, you know, these decisions aren't difficult to make, because no one in their right mind would have tried to make a few kilos using this chemistry. So this came into Chiral, and I think this was Debbie Ainge that actually did this, if she's in the audience. So what Debbie found was a quick Wittig reaction, asymmetric cis hydroxylation, oxidation, and then coupling. Goes from 2% to 55% overall yield. And I think this entire work package took about 4 weeks to sort out. I think in 4 weeks, you'd have been lucky if you were through the first step of the original process. This is a sort of good example of the sort of thing that just a simple, quick experiment. And I've got the last two, then I'll stop. You look at this and you protect the properanol, and then displace, and then do the and then you have to do the deprotection. And when you quickly look at that, you think, well, there's a big dissonance in acidity between the hydroxy and the NH. You know, perhaps you don't need to do that to protection. And you're right, 90% yield, 1 step, done. And that's, you know, that's 2 or 3 hours work. And that could be done at any stage during the LO period. You don't have to wait to the end. And I'll leave, this is my favorite example. I don't think I've ever seen one quite as good. So this was a compound, I think this was Avalon in Bristol. And you've got a demethylation followed by making a triflate and then a borane coupling to put the ethyl group from methoxy then to ethyl. But someone just looked at this and looked at it in a way to say, you know, with these 2 electron withdrawing groups and it's a naphthalene, it's not particularly aromatic, is that looking like a vinylous carbonate? And if it is a vinylous carbonate, can't you just displace this? Just stick this, would this group be displaceable? Well, as you say, it's 1 step to fight, and lo and behold, yes, you can do that 90% right off the bat. And I think in an afternoon, we'd pretty much made the tox supplies for dose range finding, and I think that it speeded up the nomination by about 4 months. So these are some good examples, but they're not unique. And if you talk to someone like Dave Ennis at AZ and look at some of the other ChemDev papers in OPRD, you can see these advantages are there all the time. I think the problem is if you get too pressured, you basically grab, it's called the bird, John Dixon used to call it the bird in the hand syndrome. You just grab what you know works and you just stick with it. You just got to be brave. So that's where I'll end. Thank you for your attention.
All right, thank you very much for that talk, Dave. I thought that was really interesting. Really cool stuff in there. I quite like the, if you have a zany idea, then try it. I reckon you can definitely get a lot from that. All right, yeah, well, I think, go ahead.
No, it's all right. No, you're right. It depends. You've got to create the right culture in your organisation to do it.
Very true. Never a truer word has been spoken, I reckon. All right, so I will pass over to Ollie and Fred, who are moderating the Q&A, if I may.
Hello, Fred. Are you on the line?
Yes, I'm here, Oliver. Yes, there was one question I noticed in the list that stood out to me, Dave. It's from a Chris Brown. There's no affiliation, and he asked about the impact of continuous flow chemistry at the interface from medchem into chemical development.
Yeah, I think it has a role to play. I think it's not going to be. I mean, funny enough, I was talking with someone in Big Pharma about this recently. I think it can have, you know, so certain things like low temperature anion chemistry can be done more effectively doing that. I think certain things, I know ozonolysis and stuff like that can be done. It can sometimes, all it'll be, in my view, there will be an extra golf club now in the bag. The same with large-scale chiral chromatography, that I thought maybe at one stage would significantly change things, but it ends up that it does some things really well and other things not quite so well. So you hear a lot from certain groups around end-to-end continuous flow, and you just put in your compound at one end and your drug product comes out the other end. Maybe it's because I'm getting old, but I just don't buy it for most compounds. In terms of a control strategy, in terms of control of purity, which is what the FDA is more interested in, I think that's probably not going to be, certainly if it happens, it won't be in my lifetime. But I think early on, certain chemistries, yeah, you can do that.
So I think when people are putting in their questions, can they state if they'd like to state their affiliation and also where they're dialing in from, because it'd be quite nice. I think we've got some people from the US. There's quite a lot of people from Europe and stuff like that. So it'd be great to hear where you're dialing in from. I've got a question about was it Berlinta synthesis? You used chlorine gas. I just wondered if you could comment on sort of scalability of that, I guess.
That's fine. You know, there are certain regulations you have to adopt to use chlorine safely on plant, but you can do it. I mean, you know, photochlorination of toluene is done on gargantuan scale. So there will be regs in terms of you're putting it into the plant in terms of chlorine cylinders, control of that. But it's a very scalable. Sometimes you end up having to use NCS because you haven't got the permits to do it. And but actually, if you can get it, it's cheap and it's ultimately very stable. So
what actually is the process? Do you just do it in a hydrogen vessel, or do you just sparge it through the
You
salt?
can do. It depends. You'd have to look at the materials of construction. But you often do it in a glass lined. You can, but it depends on the pressure, whether you have to have a pressured vessel or it's just bubbling chlorine. In that case, you just bubble it in and you scrub the excess gases coming out.
Sounds good fun. I'm not sure my managers would be very happy if I suggested that at AZ now.
Well, in process, they wouldn't mind, I'm sure.
I had a question, Dave. I was just wondering, you talked about the physical form fairly early on, but you didn't mention particle size distributions. When does particle size distribution come into play?
It depends on what your class is. So the first thing to know, I think, particle size will come into, for example, if you've got an inhaled compound, so it's going into a nebulizer or a powder inhaler or something like that, then particle size is important. But if it's a BCS class 1, you know, the chances are early on it's probably not that important. It's only when you start getting into tablets, and it again depends on how solid those crystals actually are. And I've seen situations where we haven't had to worry about any particle size. As you get less soluble and you get to BCS class 3 and 4, then particle size can be. And so you might have to add an additional particle size reduction step, either milling or through a jet mill or maybe micronization to get the particle size. It's something some people use for clinical trials to get better exposure if it's dissolution controlled, to get better exposure and get higher blood levels in a tox test. But it really does depend on it really does depend on the solubility, permeability of your compound as to how important it is.
Okay, we've got a question from
we've got a question
Martin from Australia. You mentioned trifosgene suppliers. How does that go, shipping and scale?
You can get it. So I've just used phosgene, and I don't know exactly where they got it. I think I say trifos. I thought it said thiaphosgene. Thiaphosgene is actually a bit more difficult. I think you can actually ship it. There's maybe a scale limitation. You might have to come by boat rather than air freight. And so I think that, again, you see, we're talking about time delays. That's a really good point, because if you've got something, you can buy it, but then you find that actually you do the hazard rating of this intermediate that you thought was going to be in your plant on Tuesday, and then someone's realised that actually it has to come by sea because you're not allowed to air freight it because it's too much of a potentially explosive. So now you've built in 6 weeks in terms of delay because it has to. So I think trifosgene can be used. It's a solid. I don't know. I think for a kilo or two, I'm pretty sure it was air freighted. But sometimes you can freight, and in companies that will chill, certainly you can freeze it and ship these things or fly these things. But again, at the moment, I guess nobody's flying. So, maybe I'll have to come via boat. So there are various hazard regs, UN regs, and good shipping companies will all have them. So if there's any doubt, go and contact those and get absolutely, correct information.
So why is it that thiofosgene is harder to ship than phosgene? I would have thought it would be the other way around.
No, not trifosgene. No, phosgene you can't. I don't think you can't really ship now. I think you have to generate trifosgene. I think now thiofosgene I know gets made on scale in Seal Sands up in the northeast, but they make it and use it. I think you can ship small amounts of thiofosgene. I don't know whether you can actually air freight, say 20 kilos or something like that. That might have to come by boat. But trifosgene would be safer.
Yeah.
We have a question in from James Good from Revival in the UK. He asks a question on particle size. Could you related to that? Could you, Dave? Could you tell us about the importance of polymorphism assessment and when it starts to really occur in medchem or chemical development?
I mean, you can do this sort of thing on a few tens or maybe a couple of hundred mgs. I think the way that I would look at it, the way I've always looked at it, first of all, try and get a basic assessment of solubility. So if the compound's really soluble, if it's a BCS class 1, so it's soluble and permeable, and the chances are, even if it adopts a different polymorph, then you're probably going to be okay. It's going to be soluble enough. It's permeable enough. The first thing to do is to check whether the compounds that you've got initially are crystalline, because the difference between amorphous and a crystalline form of a certain compound can be, an order of magnitude, maybe even more. I've seen huge differences in solubility between amorphous powders and crystalline solids. If it's a crystalline solid and it's a BCS class 1 and it's polymorphic, I would think you can. I'm sure some people are going to disagree with this, and regulatory-wise, it might be a bit of a stretch. But I think as long as you can get exposure, and if it changed form afterwards into the clinic, but you've got adequate exposure, I think you could figure a way. You might have to do a quick bioequivalent study. Where it becomes really important, I think, for these compounds that are pretty insoluble. So if you've got a compound that's just barely soluble enough, and all of a sudden it finds another polymorphic form, that you're in serious trouble. It was just about soluble. If it drops down, a kilocalorie per mole or something like, it's probably out of kilter now. It's just not going to be soluble enough, and not necessarily for the dose. As I said before, getting solubility in tox studies can be a real issue. I mean, there are now a lot more technologies that we can use than were around 20 years ago. And the other thing is, you really don't want to have to get the situation where the thing actually changes form halfway through production. So then you've got a real major problem there. So you can actually develop amorphous compounds. Some people may know or may not know that Crestor actually is amorphous. It's an amorphous calcium salt. The obvious issue there is you've got something that's amorphous, and for a fact that thermodynamically it's right up the top. And if there is a crystalline form, you've just not found it. Eventually it will find. In fact, there is a crystalline form of Crestor that we found about a year into production. A couple of sleepless nights. It turns out kinetically it's so slow we never actually see it in routine production. But that could have been an absolutely unmitigated disaster for the company if that actually had been. We couldn't control that. So I would say by the time you come into candidate selection, you should start doing more detailed studies. But you can do on a graph like that, maybe even less, and find out, at least have a reasonable chance of finding out if there is a form forcing it to come out. Use pure material though. You've got to use material as pure as you can.
So is this not going to be a problem if you? So I guess you mentioned earlier on about how you don't want to do a full polymorph screen, but if you're going to be going into formulation, is that not going to cause problems if they're using solvents that you might start making solvates and stuff like that? You might get a different polymorph out at the other end. If you don't have all of that knowledge, is that going to be a problem?
So by the time you've come out, so by the time you'll have come out of and you've got full-blown candidates, so the time you've made the first sort of 50 grams, 50 to, or the dose range finding, you will have actually hopefully be on the way to or getting a full-blown polymorph assessment done under that. So by the time you go to GLP tox and phase one clinical, you will have done your due diligence. But again, you see, half a gram or so, and you can find that all out. You could have front loaded a lot of that stuff. It doesn't take a long slurry experiments and quick XRPD. You'll be up, and DSC. You'll be able to figure those things out really quick.
We have a question in, Dave, from a Yannick Boers. I hope I'm pronouncing that correctly. No affiliation. He asked an interesting question, which I'm sure a lot of sourcing managers would not want to answer. He says, what are the best websites to use to get good sources of raw material pricing?
Oh God, I don't know. I use SciFinder, and then I call up colleagues that I have as sourcing agents, and I use those. I don't know how to. I mean, it's an absolute minefield. All of us go on SciFinder and they see, one company listing 55,000 compounds, none of which they've made, and all have a 6 weeks lead time. It is just a while. So I have a few personal contacts who operate sourcing businesses in India and China, and that's, if it's something tricky, then I will ask those to have a look. Most of the good CROs around have some pretty good sourcing departments. So if you're worried about scaling chemistry, the CRO that you go to, if you're going to use a CRO, I think they are the best. But I think this is a bigger mind. This is like the Wild West, and most of it is finding suppliers you actually trust. So that's, even if it's on a website, it's not, how reliable that is, and is that the actual place that makes it? I don't think you get any chance of figuring that out.
Well, we've had a follow-up from Chris Brown, who's at Evotech in the UK. I think I've met Chris, and he asked quite a broad question. But what would be your go-to analytical methods for measuring purity?
Well, it's going to be a cheat answer. It depends on the molecule. Depends on the technique. Guides the technique. So, for example, if I had a CF3 group and I wanted to know, I might do an assay based on fluorine NMR. Limit of detection on fluorine NMR is, I think we did a method for trifluorite, GMS triflate, and I think we could get down to parts per billion in terms of quantifying by NMR. By and large, I would always tend to believe NMR over. It depends on the level to which you need to know that answer. So obviously the advantage with NMR, you see everything. You don't miss anything, and that's why it's not easy to get a definitive answer. What is the purity? It's all about relative purity. That's why a full spec takes so long to test. You do your relative subs with HPLC. You do GC to get residual solvents. You do sulfated ash or ICP-MS to get inorganics and heavy metals, and there could be other tests that you do, and then you put them all together and you say, okay, on average that looks around that pure, but it's purer than that. To actually get to the stage of absolute impurity, it's impossible to do, because you don't know the response factors for the low level impurities. That's where NMR comes in. So if you've got a relatively simple molecule or really stand out in an internal standard, I would, for a quick and not particularly low limit of detection, but not missing anything, I think NMR would be my go-to initially. So
do you think that strength assays should be introduced a lot earlier then? considering they're not really used that much in medchem at the earlier sort of
I
stage.
think it depends. Yeah, it depends on what decisions you're going to be making. If you say, right, this thing's a bit unstable, we're not going to use it. We're going to deprioritize it. Then just make an effort to know, how pure is that? I'm sure most people do, but sometimes, or you've left the thing lying around for a while, it's decomposed a little bit because it's not particularly stable. And then you get this folklore, oh, we made that, but it's not stable. And then you'll make it again, you get it clean, it crystallizes, and it's as stable as bricks. That's the thing, is what decision you're going to be making based on those data. If it's key, quite strategic, then do the bloody thing properly.
All right, one last question. That's probably all we have time for. But I was just wondering about, you were saying 20 to 30 grams for a full salt and polymorph screen. I mean, I work in high throughput, so I'm normally dealing with tiny amounts. What do you get for 20 to 30 grams? It sounds like a lot to me.
You would. I mean, you'd get a lot of it is stress testing, and what you will get a chunk of for that, what you should do, is get a reasonable scale, say 10 grams or so, maybe a bit more, in terms of crystallization development, a production method that will enable you to produce that polymorph that you've selected. Because until you've got that, you haven't got anything. That's where a lot of the material comes. So it's confirmatory, you're doing a lot more stability, and you're looking to push these various forms and see which one, in some cases it's very obvious which ones are thermodynamically most stable. In other cases it's not so obvious. And then you've got this balance between, is it thermodynamically it's the most stable, but it's not chemically the most stable. And if it was, if it's not the most stable, would you risk actually going with it or not? You may decide that actually for the first in man, it's a risk we'll live with. And to be honest, it's a risk we're all living with, because actually it's one of the few things that you can't actually know when it's going to change. You can do your due diligence and try and force out the most thermodynamically stable. I mean, think of this, Trevor Laird was telling me, I think there was a new form of paracetamol about 4 or 5 years ago, and it can just come out. I mean, Avandia changed form at tech transfer. I had a friend of mine working in the labs at Tunbridge, and he was getting phone calls from the CEO, starting with a very encouraging face, what the F's going on? It's that, it just came out of the blue. And what happens is you get things that are cleaner and cleaner, and you're removing these habit modifiers, small amounts of impurities that get in the way, and then template for less stable polymorphs. And one day they disappear, and all of a sudden the true nature comes out. But if you think, I think Guy Lloyd-Jones was telling me, it's about 50 molecules can get together to start a nuclear crystallization. Some of these habit modifiers can be well below the limit of detection, so you'd never really find out what they are.
That makes sense. It's quite a scary thought, really.
Don't think about it.
Yeah, I know, right? I think we are pretty much at time, so let me back to this. So, thank you very much for your talk, Dave. Thanks so much for helping out with moderation to Ollie and Fred. I thought that was a really fantastic webinar. So next week we have another from Mike Waring at Newcastle University. He's going to be talking about approaches to hit finding using fraglites and encoded libraries. And he's doing a joint talk with Simon Peace from GSK, who is talking about development of facile and flexible 2+2+2 cyclotrimerization methodology and its impact on 2 GSK lead discovery programs. And lastly, thank you to all of you for listening in. Please join us again in the future.